Good day, everyone, and a very warm welcome to our session on ICH Guideline Q8, Pharmaceutical Development. It's great to have you here as we dive into one of the most influential guidelines shaping modern pharmaceutical quality, design, and innovation. Today's discussion will help us explore how Q8 encourages a deeper scientific understanding of products and processes, and how concepts like quality by design, design space, and enhanced development approaches can transform the way we think about pharmaceutical manufacturing. I'm pleased to introduce us. My name is Peter, and with me is Monica. We will guide you through the key principles, practical implications, and real-world applications of Q8. So, settle in, get comfortable, and let's begin our journey into the heart of ICH Q8, where science, quality, and innovation meet.
Whether you are prepping for a high-stakes regulatory meeting or maybe catching up on the latest structural shifts in the pharma field, or, you are just insanely curious about how things are made behind closed doors, we've got something special.
We really do.
Our mission today is to unpack a truly foundational document from the pharmaceutical world. We are looking at the ICH Harmonized Tripartite Guideline on Pharmaceutical Development, specifically the document known as Q8R2.
Which I know sounds incredibly dense on the surface.
Oh, absolutely. A bit of alphabet soup.
Right, but the implications of this document are just profound. It completely rewrites the rules of engagement between pharmaceutical manufacturers and regulatory bodies worldwide.
And to give you a sense of why this is such a big deal, we really need to look at the traditional paradigm it disrupted. Because historically, manufacturing medicine was essentially following a rigid recipe.
Exactly. It was empirical, trial and error.
Right. You made a batch of your drug, and then you tested the final product at the very end to see if it met specifications. And if the batch passed the lab tests, you shipped it out.
But if it failed?
If it failed, you threw millions of dollars of inventory straight into the trash. But this document establishes a massive structural shift. There is a line right in the text that captures this revolution perfectly. It says quality cannot be tested into products, i.e. quality should be built in by design.
What's fascinating here is how that single sentence completely changes the entire philosophy of manufacturing. It moves the industry away from that empirical trial and error to a predictive, science-based approach.
Which is huge.
It is. This isn't just another regulatory hurdle. It is actually a framework that allows for what the guideline calls flexible regulatory approaches.
I mean, they get more freedom.
Exactly. Historically, if a manufacturer wanted to tweak a process parameter, maybe adjust a drying time or a mixing speed, they were just buried under post-approval paperwork and regulatory submissions.
No nightmare.
Right. But Q8R2 introduced the concept that if a manufacturer can prove they deeply fundamentally understand the multivariate science of their product and process regulators will grant them the operational freedom to innovate.
And optimize without that constant oversight.
Precisely.
Okay, let's unpack this.
Yeah.
Because getting that kind of operational freedom from a regulatory body obviously requires providing a very specific, highly rigorous blueprint of the drug up front.
You can't just ask for it, you have to prove it.
Right. And the document relies heavily on two foundational concepts to build that blueprint, the QTPP and the CQAs. So let's look at how those are defined in this framework.
Okay, so the QTPP stands for the Quality Target Product Profile.
Which means what exactly?
You can think of this as the basis of design for the development of the product. It starts with the clinical end goal in mind.
So what the patient actually needs.
Right. It considers the route of administration, the dosage, form, the exact strength of the medication, and the delivery system. The QTPP is the definitive profile of what the patient requires.
And to guarantee the product actually meets that clinical profile safely and effectively, you have to establish the critical quality attributes, or CQAs.
Yes, the CQAs.
The text defines a CQA as a physical, chemical, biological, or microbiological property or characteristic that absolutely must be within an appropriate limit range or distribution to ensure the desired product quality.
And the guideline provides specific examples that illustrate how widely these attributes vary depending on your product profile.
Like it depends on what you're making.
Exactly. For a solid oral dose, like a standard pill, the CQAs are typically purity strength and drug release or dissolution.
How fast it dissolves in the stomach.
Right. But if your QTPP dictates an inhaled product, your CQAs shift entirely to focus on aerodynamic properties.
Because it has to reach the lungs.
Exactly. For parenterals or injections, the non-negotiable CQA is sterility. And for transdermal patches, you must tightly control adhesion properties.
so it actually sticks to the skin. The text also points out, though, that these attributes don't just apply to the finished product. The raw drug substances and intermediate materials have their own CQAs.
I do.
It mentions things like particle size distribution and the crystal properties of the raw active pharmaceutical ingredient.
And the rationale there is that the CQAs of the raw materials cascade directly into the CQAs of the final drug product.
Right.
If you don't control the polymorphism or the crystal shape of your raw powder, it really doesn't matter how well you compress the tablet. You will likely fail your final dissolution targets.
Because the foundation is flawed.
Exactly. Identifying every single one of those potential attributes from the raw input to the final output is the very first step in building quality by design.
Here's where it gets really interesting, though. Once a manufacturer locks down those required attributes, knowing exactly what the dissolution rate or the aerodynamic properties need to be, how on earth do they trace back which of the thousands of variables in a manufacturing facility will actually get them there?
Right, it's a massive puzzle, and it requires a massive cross-functional undertaking very early in pharmaceutical development. A team of experts sits down to perform a comprehensive risk assessment.
So they don't just guess.
No, not at all. They pull together all prior knowledge, literature, and initial experimental data, and they use formal risk assessment tools.
Like what?
Well, one prominent tool mentioned is failure mode effects analysis, or FMEA.
Okay.
They systematically score every single process parameter and material attribute based on 3 distinct factors.
Which are?
The probability of it going wrong, the severity of the impact on a CQA if it does go wrong, and its detectability.
Detectability meaning, do we even have the analytical capability to notice if it drifted out of specification?
Exactly. If you can't detect it, it's a much higher risk.
The document provides a fantastic visual example of this in the appendix using an Ishikawa diagram, which is commonly known as a fishbone diagram.
A classic engineering tool.
Right. It maps out a standard granulation process. You have the main spine representing the critical quality attribute you are trying to protect, and the ribs coming off that spine represent the broad categories of inputs that could cause a failure.
And those ribs get incredibly granular. On the raw materials rib, the team evaluates the particle size of the drug substance, the specific grades of diluents, the binders, and even the water quality.
Wow, just on that one rib.
Yeah. Then on the drying rib, they scrutinize the temperature, the relative humidity, and the airflow rates. Then you have the compressing phase, where the actual machine variables come into play.
Like feeder speed, press speed, and punch penetration depth?
Precisely. Even environmental plant factors and operator techniques are mapped as potential sources of variability.
Let me make sure I am visualizing the operational reality of this correctly. You have feeder speeds, moisture content, and punch penetration depths all interacting simultaneously.
Yes, all at once.
You can't just test one of those in a vacuum to see if the tablet holds together.
No, and if we connect this to the bigger picture, this multivariate complexity is exactly why the old trial and error method is completely obsolete.
Because there are too many moving parts.
Exactly. You take this massive, prioritized list of high-risk variables from your Ishikawa diagram and your FMEA, and you apply formal experimental designs.
Which the industry calls design of experiments, right? Or DOE.
Right, DOE. Instead of the archaic method of testing one factor at a time, you systematically test how these variables interact with each other.
So testing combinations.
Yes. You might discover that a high drying temperature is perfectly acceptable, but only if the initial moisture content is below a certain threshold.
Okay.
If both are high, simultaneously, their interaction causes an unacceptable degradation of the active ingredient.
Which brings us to the absolute holy grail of this entire document, the design space.
Yes, the design space.
The guideline officially defines the design space as the multidimensional combination and interaction of input variables and process parameters that have been demonstrated to provide assurance of quality.
It is essentially the mathematical territory where all those interactive variables perfectly align to guarantee the product meets its CQAs.
And the regulatory payoff of establishing this cannot be overstated. We mentioned the reward of operational flexibility earlier. The golden rule established by the Q8R2 guideline is this: working within the design space is not considered a change.
That is the most critical sentence for manufacturers.
Right, so to translate that to the operator on the floor, if the approved design space dictates a complex relationship between temperature and humidity and a sudden weather event, drops the ambient humidity in the facility.
The operator has permission to instantly crank up the process temperature to compensate.
Exactly. As long as that new combination falls within the established mathematical boundary, they don't have to pause production and file a deviation report with the regulators.
They just keep running.
Yeah.
But movement out of the design space is considered a change, and that triggers a regulatory post-approval change process.
It's like a hard stop.
Right. The regulators are essentially saying, prove to us the exact boundaries of your scientific understanding. As long as your process stays within those proven boundaries, you have the autonomy to operate and optimize continuously.
The visual math provided in Appendix 2 of the text to explain this is just brilliant.
It is an excellent use of data visualization.
You get pretty excited about the math here.
Oh, I really do. It's fascinating. The text uses contour plots and 3D response surface plots to demonstrate how a design space is calculated from the DOE data.
Okay, walk us through it.
Imagine mapping out two CQAs for a solid oral dose. Dissolution, which is how fast it dissolves, and friability, which is how easily the tablet chips or breaks.
Because nobody wants a bottle full of broken tablet dust.
Right, so friability has to be tightly controlled alongside the dissolution. You are trying to optimize parameter 1 and parameter 2 of your manufacturing process, say the granulation fluid volume and the mixing speed.
Okay, I'm picturing the graph.
The plots show how these two process parameters interact to affect both outcomes. If you require dissolution to be above 80% and friability to be below a certain percentage, the design space is the specific overlapping area on the graph where parameter 1 and parameter 2 meet both of those goals simultaneously.
And the text notes that this approved space can take different shapes depending on what the manufacturer actually requests.
Yes, it can be very straightforward or very complex.
Right. It can be a simple linear box. Like for sheer operational simplicity, a company might submit a space that says parameter 1 stays strictly between 44 and 53, and parameter 2 stays between 0 and 1.1.
Just a simple rectangle on the graph.
Exactly. But it can also be a complex non-linear curve to give the factory maximum operational range. The mathematical example given in the text for a nonlinear space shows that if parameter 1 is at 46, parameter 2 can swing anywhere between 0 and 1.5.
Wide range there.
Yeah, but if parameter 2 is fixed at 0.8, parameter 1 is restricted to between 43 and 54. So the acceptable range of one parameter is constantly dynamically shifting based on the real-time value of the other.
And it becomes even more complex when you consider that a design space doesn't just apply to fixed parameters. It can be entirely time-dependent.
And wait, time-dependent?
Yeah. The guideline highlights the example of a lyophilization cycle, freeze drying. For lyophilization, the design space is an actual trajectory over time, charting product temperature and chamber pressure over a multi-day cycle.
So it's a path you have to follow.
Exactly a path. If the process strays above the upper limit of that established path, the text warns of excessive impurity formation. If it drops below the lower limit of the path, you risk excessive particle attrition, meaning the high-value biologic product could be completely compromised.
That is an incredible level of precision.
It really is.
Now that we have established the invisible mathematical boundaries and the process parameters, let's look at how the guideline treats the physical components going into this design space.
Because it's not just about the active ingredient.
Right, the active drug substance is the obvious focus, but the text is remarkably strict about everything else surrounding it.
The quality by design framework requires a rigorous justification for every physical element. For instance, the guideline specifically addresses the practice of using overages.
That part really stood out to me. The text points out that using an overage, which is the practice of adding extra active drug substance into the batch to compensate for expected degradation over its shelf life, is actively discouraged.
Yeah, it's heavily frowned upon.
Why is that?
It is discouraged because relying on an overage usually masks a fundamental lack of process or formulation understanding.
Like a Band-Aid.
Exactly. If a manufacturer is simply adding extra active ingredient, hoping that enough survives until the expiration date, they are not actually controlling their quality.
That makes sense.
So the guideline mandates that if an overage is used, it must be fiercely justified concerning the safety and efficacy of the product. The manufacturer has to document the exact amount, clearly identify the specific reason for the manufacturing losses or degradation, and prove that the excess won't cause adverse effects if it doesn't degrade as expected.
Because if it doesn't degrade, the patient gets a higher dose.
Exactly, which could be dangerous.
And then you have the excipients. The manufacturer has to prove that these inactive ingredients, whether they are antioxidants, penetration enhancers, or disintegrants, provide their intended functionality and perform consistently throughout the entire intended shelf life of the drug.
And that holistic requirement extends heavily to the packaging as well.
Oh, right, the containers.
Under Q8R2, container closure systems are treated as a highly engineered component of the drug product itself. The packaging is an active barrier protecting the drug from light and moisture over years of storage.
It's not just a plastic bottle.
It's not at all. The manufacturer must extensively study sorption, which is the risk of the active drug sticking to the inside of the container and lowering the actual dose the patient receives.
Oh, wow. I didn't even think about that.
Yeah, and they also have to test for leaching, where the chemical compounds from the plastic or glass container actually seep into the formulation.
And this gets exponentially more complicated when the packaging includes a dosing device, right? Like a pen injection device or a dry powder inhaler where the packaging is literally the delivery mechanism.
Yes, that's a whole other level.
The text states it is critical to demonstrate that a reproducible and accurate dose is delivered under testing conditions that simulate real-world patient use as closely as possible, accounting for things like patient inspiratory flow rates.
Yeah. And when discussing physical components and shelf life, the guideline also places strict parameters on microbiological attributes.
Right, the preservatives.
For multi-dose products that aren't inherently sterile, but contain an antimicrobial preservative, you cannot simply flood the formulation with preservatives to guarantee safety.
Which would be the easy way out.
Right. But the text requires manufacturers to use an antimicrobial effectiveness test to determine the lowest specified concentration of that preservative that remains effective at controlling microorganisms throughout the product's entire shelf life.
So what does this all mean? We have defined the ultimate clinical goal with the CQAs. We've mapped out the multivariate risks on an Ishikawa diagram. We've calculated the perfect multidimensional design space using design of experiments.
There's a lot of work.
Yeah, we've rigorously justified our excipients and engineered our container closures. So how does a facility ensure that the day-to-day, minute-to-minute manufacturing actually stays inside that perfect mathematical boundary?
Well, that is the function of the control strategy.
Okay.
This represents the ultimate culmination of the shift toward quality by design. In the old empirical model, the control strategy relied heavily on end product testing to make a final batch disposition decision.
Testing at the very end.
Right. But quality by design allows a manufacturer to shift those controls upstream. Because you understand your sources of variability and their interactions, you can adapt and control the process in real time.
This introduces a concept that completely revolutionized factory floors. Process analytical technology, or PAT, and the capability for real-time release.
Yes, PAT is the enabler for that upstream shift. Instead of waiting for a batch to finish quarantining the inventory and waiting weeks for a laboratory to run high-performance liquid chromatography tests.
Which takes forever.
It does. PAT involves measuring critical quality attributes inline or online during processing.
While the machine is running.
Exactly. The guideline provides excellent examples of how this changes testing. For instance, instead of running a lengthy traditional dissolution test on a fast disintegrating solid pill at the end of manufacturing, you might use a rapid disintegration test during the process as a surrogate.
But only if you prove they were linked right.
Right, provided your experimental design definitively proved the two are linked.
The most striking example from the text is how they handle content uniformity testing to ensure every single pill has the precise required dose.
Oh, this is brilliant.
Under the old framework, you'd pull a statistical sample of finished pills and run them through a lab, leaving millions of dollars of product sitting in a warehouse waiting for approval. Under this framework, they describe using continuous weight variation checks coupled with a near-infrared or NIR assay directly on the manufacturing line.
The implementation of near-infrared sensors scanning the materials continuously as they move through the equipment is just a massive leap forward.
Scanning every single pill.
Yes. Because you are confirming both the weight and the chemical composition simultaneously in real time, it allows for what the guideline calls real-time release testing.
So no lab wait time at all.
None. This can completely replace traditional end product testing. And as the text points out, this methodology actually provides a significantly higher level of quality assurance.
Because you aren't guessing based on a sample.
Exactly. You are monitoring the entire batch continuously as it is being formed, rather than just sampling a tiny fraction of the final product and assuming the rest of the batch is identical.
The journey of a pharmaceutical product, as dictated by ICH Q8R2, is a masterclass in risk management and systems engineering. I want you, the listener, to keep this in mind the next time you hold a medication.
It really changed your perspective.
Realize that it is not merely chemicals mixed in a vat. It is the culmination of multidimensional mathematical modeling, rigorous risk assessment, mapping out every conceivable multivariate interaction, and a highly controlled design space monitored by real-time analytics, all tailored to guarantee that the product works flawlessly every single time.
This raises an important question, and it is something worth reflecting on. The pharmaceutical industry has demonstrated that by leveraging deep scientific understanding and multivariate risk assessment, it is possible to map multidimensional design spaces that guarantee the quality of highly complex products without ever having to rely on end product testing.
Which is incredible.
It is. If this framework can be successfully applied to life-saving biologics and complex pharmaceuticals, what other complex systems in our world could be radically transformed by this philosophy?
That's a great point.
Imagine applying this predictive rigor to our global food supply chains, the construction of our critical infrastructure, or even the architecture of our digital networks. What would happen if we stopped testing for failures at the end of the line and truly shifted our mindset to building quality in by design?
That is a phenomenal concept to leave you with today. Thank you for joining us for this deep dive into the blueprint of modern pharmaceutical development. We hope this exploration provided the shortcut you needed to be well-informed and perhaps sparked a few fresh perspectives on how complex systems operate around us. Until next time, keep exploring the details that shape our world.